right, it's 11:31. We'll get started. I'd like to start by acknowledging the traditional owners on the lands on which we're meeting today and pay my respects to elders past and present. Welcome everyone to the CCGCM lecture series. We've got a fantastic speaker today. I'm very excited. It's Associate Professor Paul Ekert, who's a clinician scientist. You can tell by the shoes.
Because they're expensive.
The appropriate amount of casual, but so in all seriousness, Paul is a group leader at CCI. He's a translational genomic lead for the Zero project. I've known Paul for a long time now. He's easily, my go-to person for paediatric cancer genomics. He gets very excited about cancer genomics, like we all do. So it'll be great to hear from him today. He's speaking about the Zero program, which, in my opinion, is probably the flagship cancer program for sequencing of cancer genomics in Australia at the moment, and I'm very excited to hear about it. Thanks, Paul.
Can applaud at the end, depending on how I go. That's okay. Thank you very much for the invitation. Right, I'm going to just get myself orientated. It's a real pleasure to speak to this meeting. I apologise if anyone was at the talk I gave yesterday, because there'll be quite a bit of overlap with that. But what I want to cover today is some of the background and achievements of the Zero Childhood Cancer programs. I'll give you some historical context there, point out some of the changes we've had to make this program really run in Australia and to make it successful, and some of the outcomes directly from that. I then want to sort of divert a little bit to some more biological approaches, and I hope that that's appropriate for this meeting, because one of the things that we're finding that much of what we see in cancer genomes, whilst really interesting, we don't understand, but these are real therapeutic opportunities that we have a chance to cover. And I'm going to talk a little bit about some of the findings for germline risk, which I think are very interesting and certainly very worth considering as these genomics programs roll out. And I'll touch on some of the sort of side hustles in the Zero program for you to consider. So the purpose of the Zero program, when it set out, was first to test the feasibility of a genomics program in paediatric cancer, and to expand the capacity for any kind of gene testing for children with cancer nationally. We had an ambition, of course, to improve access to targeted drugs that might otherwise not be available to those children, and to, in doing that, improve cure rates and minimise impacts of therapy. But very importantly, and particularly this is part of my role within the program, was to maximise the research and translational impacts, because we are now in a position where we sequence every child with a cancer diagnosis in Australia and are amassing an enormous amount of both clinical and genomic information on that cohort. And this is a real opportunity to discover new biology and potentially new therapy for those patients. So it started in 2015. That's when my involvement started there. I was at the Murdoch Children's Research Institute at the time, trying to get an RNA sequencing program in ALL off the ground, which we did manage to do within that single institute. And some of what we learned from that program was directly translatable into the pilot study, which took about 2 years, and we did maybe a bit more than 50 patients. It says 50 up there. We did maybe closer to 60 children over 2 years, almost all from Sydney Children's Hospital in Randwick. And in that, we were building our capability. We were trying to work out what sequencing platforms and technology we would use. We were building the whole capacity of the Zero program, making our mistakes along the way. And then that rolled out in 2017 to the national program, where we targeted high-risk children. Now, what high-risk cancer in childhood means we defined as a survival of 30% or less predicted at enrolment or at diagnosis, or relapse and refractory disease at the time of enrolment. At the end of that pilot study, well over 400 children had been recruited. In fact, we would think we captured every child in the country with that 30%, with that high risk category. And then we were funded through the MRFF to expand that program to all children with a cancer diagnosis, which rolled out progressively from 2021 onwards. And that's been a really interesting journey because we know the impact, and I'm going to show you the impact of this program in high-risk cancer. It's a much more challenging question to know how much genomic analysis can impact diagnosis, prognosis, treatment outcomes for children with standard risk cancers. If you think, for example, that the commonest childhood cancer is BALL, and a standard risk BALL has a survival rate of somewhere between 85 and 90% in this country. So are we going to improve that significantly? We might reduce side effects potentially if there are targeted therapies, but that's a bigger challenge to know whether we're going to impact in that cohort. You can see on this side here that we have a number of other programs. I'm going to touch on this high throughput screening program briefly during the talk. So some of the things that we had to discover and had to learn along the way, we committed ourselves to whole genome sequencing and whole transcriptome sequencing. In the early instances of this program, we did deploy some panels, actually one of the Peter Mac panels from Stephen Fox, and we saw no net gain in panels. We never really reported any findings off panels in childhood cancers, and that shouldn't be a surprise because all of those panels were designed with adult cancers in mind. And so it would be the serendipity of the same mutations that happen to be more common in breast cancer or ovarian cancer showing up in childhood cancers. They were focused on point mutations, not on structural variants, for which pediatric cancers are heavily biased. So we had committed to doing whole genome, whole transcriptome sequencing off fresh tissue, not off FFPE. So we don't do any FFPE samples within the program. And Sean knows this. So what did that entail? That entailed having champions for Zero at every centre. It challenged the sort of regimen of sample of biopsy and what happens to those biopsies in theatres. So we had to work incredibly hard and collaboratively to get that to work, but it does work. We are fortunate in that all children basically with a cancer diagnosis are treated within the public system. So they're treated within 9 centres. There's not that sort of diversity that exists in the adult cancer world. And so we really, if we could capture the hearts and minds of those 9 centres, we could get those samples fresh to us. And that process now works extremely well, works in every one of the 9 centres that treats childhood cancers around the country. And we, for our purposes for childhood cancer, can see no real point at this point going backwards and trying to reinvent FFPE, whole genome and transcriptome sequencing, at least for this cohort. Let me go back there. We had to build a multidisciplinary curation team, which simply did not exist at the time of the program. That expertise was not present in the country. The people who knew what sequencing data looked like and how to turn that into an assessment of pathogenicity of structural variants, of deletions, of mutations didn't exist, particularly not within the pediatric cancer domain. And so that whole program was built from scratch, and now there are about 22 people in all centres around the country. They are online twice a week, sharing all of that data through a platform we call Zero Dash, which is a sort of one-stop shop where all of this data can be collated. And so that has built, a whole infrastructure that supports the program, and it's been incredibly successful. And it's been a vehicle for education for our oncology trainees and fellows, which is quite let's say, a paradigm shift for much of paediatric oncology from where it's at maybe 15 years ago. The multidisciplinary tumour board had to be started, and this was a thing that never existed prior to this program. Most of the paediatric oncology programs in each of the capital cities of Australia kind of worked in isolation. They got together, of course, but they're not around sharing this kind of data and not around making decisions based off genomes that might affect cancer treatments. So that had to be started, and I would be fair to say there was a reasonable amount of scepticism at the start of the program. People were really unclear as to what that might do and on what basis they could make a clinical decision on given this information. That's now a weekly national hookup, incredibly well attended, and has really become part of the landscape such that people consider this standard of care within that domain now. Perhaps the biggest roadblock that we faced was what oncologists might do with this information and how they would treat it. And right at the start, and I'm not going to defame anybody here because I've said it openly to them, people would often take this information with what I, sitting there as a, helping run the curation team, would say is a gold standard. You must treat this lesion, this genomic lesion, say, thank you very much. I'll put that in my back pocket. I'll carry on with standard of care therapies because that's what they know best and that's what has given them the outcomes. And then whenever everything goes bad, I'll take that back out of my back pocket and I'll treat. So bear that in mind because that greatly influences the thinking about some of the data you're just about to see from our outcomes. And then, of course, we had some additional programs on the side that we really tried to get going. In vitro high throughput screening has turned out to be definitely feasible. It's questionable whether people ever make a decision based on in vitro testing alone. And I think we haven't answered that question yet. We attempted at the start to do as many PDX models as possible. All good? Someone's just coming into the meeting right now. To do as many PDX models as possible because the initial ambition was to try and have a mouse avatar along with most patients. That just turned out to be completely impossible to achieve. We should have known that from the start. We know it now. That program is now really focused on specific tumour types, on ones where there's not enough models around to test. But that is more or less finished in its original guise. So I want to come to the lessons that we've learned and the highlights of the program. And this relates to two papers that we published, this one in 2020 in Nature Med, and then there was a follow-up paper also in the same journal in 2024. And you can see the co-first and corresponding co-last authors there arrayed on the top. And just, a little bit of shameless self-promoting, if you don't mind. So rarely get to do this, but it got named as one of the top 10 clinical genomics advances in 2021. That's the Zero program based on this paper, because I think I'll be able to show you what what got those reviewers over the line. The high-level findings you'll see there are on the bottom. We had reportable findings in virtually every cancer that we would find, so that shouldn't surprise us, but this was a feasibility, so we wanted to know that we could do that. We made a lot of treatment recommendations. Some of those are varying tiers. Tier 1, where we had the strongest, most compelling information that something was a driver and specifically targetable, all the way down to differences in gene expression influencing treatment recommendations. We changed diagnosis in about 5%, and the rate of cancer predisposition was at that time much higher than expected, about double what had been previously reported a year prior in the New England Journal. That number's held true actually across a number of studies now. One thing to recognise about the cohort, as I mentioned, we were high risk. The commonest childhood cancer is BALL, and you can see there that we're really underrepresented for the more common paediatric cancers because they didn't fit that category of high risk. So that distribution is now changing as we enrol all patients with cancer. So our numbers of BALL and TALL rapidly expanding, and now including the more standard risk patients. You can see the range of disease states that they were from. And the sort of mutations and gene ranges, pathways that we observed shouldn't surprise anybody. These are the sort of canonical cancer pathways. The types of mutations that we saw were a little distinct from those that are more common in adult cancers. And we have a clear bias towards structural variants. Most childhood cancers have 10 to 100-fold fewer mutations than average adult cancers, somewhere between 0.1 and 1 mutation per megabase. That's the normal range that we deal with, unless those children have a congenital mismatch repair syndrome. Then that's really almost the only time we see mutation rates closer to 100 per megabase. So this was the data, I think, that ultimately persuaded the Nature Medicine reviewers and editors to accept our paper and reject the co-submitted one from St Jude's, which was a real bonus for us at the time. And that was that we had some early indications that we were having a clinical impact. So recall that these patients are heavily pre-treated with a 30% or less chance of survival. We did not anticipate we were going to make any major clinical impact on those who even received a recommended treatment. So remember, we recommended treatments in up to 70% of these patients, and 43 of 237 received a treatment, many of them quite late in the stage of their disease. So the chances that we're really going to see partial remissions or even complete remissions in these patients, we thought was vanishingly remote. But nevertheless, there were some signals here, and as I'll show you later on, many of those related to us finding receptor tyrosine kinase activating events where there are targeted treatments that are available. There were 1 or 2 Hail Marys in here, 1 or 2 immunotherapy responses that we hadn't anticipated would come through, mTOR inhibitor responses. So there were a signal here, if you like, that we were getting a clinical response. And the other thing to note here, what this is showing you, is that across all those tiers of evidence, the gold standard evidence right the way down to differences in expression were contributed to some of these responses. So this was the follow-up, and this paper was specifically about following up treatment responses where we had about a minimum of 18 months follow-up of treated patients, led by Loretta Lau and David Ziegler. And one of the things you'll notice is that the number receiving a PGT, precision-guided treatment recommendation, is about the same. The number actually getting the recommendation has doubled in that time. And again, we are seeing some objective response rates and objective benefits. But there's an interesting story to tell you here. With precision-guided treatment, overall, this is at 2 years, remember, so it's early days, and you can see the curves ultimately come together in this high-risk population. There is a trend towards those precision-guided therapies providing some benefits, at least for a period of time. More importantly, though, if those precision-guided treatments are instituted before progressive disease occurs, then you start seeing significant changes in the clinical benefits. So that idea of sticking the knowledge in your back pocket and waiting till everything goes bad is, not surprisingly, not the best way to get the benefit from this kind of information, even when it's you don't have a clinical trial, for instance, to lean on to prompt that decision. And the biggest benefit was seen when we had that tier 1 evidence, the highest possible evidence connecting gene to drug response, that it was a structural variant, almost always some kind of fusion or a deletion driving oncogene activation. And that treatment was started earlier. Again, in a high-risk population, we had an 88% response rate. Again, we hadn't anticipated that was what we were going to see. But when those three factors line up, you get the best outcomes. So that's where Zero stands at the moment. We don't know how well this is going to apply to standard risk patients. That's an open question and an important one for us to answer. We need to be able to provide the right service, if you like, for the right patients. But what we have shown across these two studies is that it's feasible to undertake this kind of genomic analysis. I think Zero remains the only instance of many genomic programs around the world, many of them very excellent, but this is the one that has worked at a national scale and is rolled out at a national scale. I think it's the only example of that, or the most successful example of that. Perhaps that's a more reasonable claim. It's required an enormous amount of work to get this going, but now it is, and everything is embedded. For us, whole genome and transcriptome sequencing provides the greatest precision and the greatest potential benefits for patients. And we argue that we see the best, that we can see, strong clinical signals when the right lesions are found, and particularly when treatment is instituted earlier in the course of disease. Now, I guess because this is the collaborative genomic session, I just want to argue for the integrated analysis of RNA sequencing within whole genome sequencing. This has become key for us. I imagine you've probably had Chelsea Mayo speak. Well, I know she's spoken here before, and she's a champion of this. But we see, considerable benefits from combining those two platforms together. It helps us give some functional context to the genomic findings, and I'm going to illustrate that just quickly here. Very often you can see genome shattering or complex genomic events that connect multiple genes together in a very complex rearrangement. And prior to the institution of things like links with whole genome sequencing data, we relied heavily on RNA sequencing data to resolve these sorts of very complex gene shattering or multi-hop events into what turned out to be a very targetable and very simple ALK fusion, an ALK rearrangement. And so we see these sorts of things over and over again, and RNA sequencing helps us resolve those. And so we argue that the combined analysis is really important. We've tried our best to improve how RNA sequence analysis can detect other events. So, for example, intragenic or intergenic deletions. Sometimes RNA sequencing can be very challenging to make those calls. We've defaulted sometimes to a reference-free approach of RNA sequencing analysis to pull these back out. We've used Alicia's Minty algorithm quite a lot, and I find that very effective. But, the expression of, for example, an IGH rearrangement that drives expression of the target gene, and you can see that in the tumour, or an intragenic deletion that actually abolishes expression of the tumour suppressor is nice orthogonal evidence to be able to deploy. We use allelic imbalance or allelic expression of oncogenes, and this is what this is showing you, the different types of mutations you might see, whether they're heterozygous or homozygous in the DNA, but the consequences in the transcriptome give you a sense of function. So if an oncogenic mutation is not expressed in the genome at all in the RNA, is it really an important driver? So, or if the wild type allele is not expressed at all in a homozygous context, that might be a more important driver. So we use that information in an integrated way to make our calls. We use methylation, so all of the samples get 850K methylation arrays, and we hardly use that data at all. We only use that in using the Heidelberg methylation classifiers, in essence. So the data sits there, but we haven't really deployed it beyond this sort of diagnostic categorization. So that works for brain tumours. That was from Stefan Pfister in a Nature paper, I think in 2018 or 20. And that's really become a standard diagnostic tool. You do the methylation array and it gives you a very close both molecular and histological type diagnosis. And they now have one available through their site for sarcoma, and there are others coming. So classification turns out to be a tool that our oncology colleagues really like to use, and it's now, I think, fairly standard. I mentioned at the start about the genomic risk of childhood cancers, and this is really work led largely by Mark Panesi at the Institute, and Noemi Fuentes-Bonos, who's a dual-trained paediatric oncologist and geneticist. So we found about double the rate of germline predisposition alleles driving childhood cancers. And there are a couple of really interesting, I think, lessons that we learned from this. The first was that whilst the association between a germline allele and the cognate tumour, that is the typical tumour that would derive from that germline predisposition allele, was dominant, though that was there. I shouldn't say dominant to a genetic audience. That's not what I meant. That was more common. About 40+% were in tumours that are non-cognate. They're not the tumour type that you expect that particular allele to be driving. So that would mean if you were doing genetic testing based on tumour subtype, you would miss about 40% of those germline pathogenic alleles. And you can see the range of those there, but the shaded versus the bolder colours there tell you that, you know, we have a significant proportion. And what it really means is that we don't understand the full extent of tumours that these hereditary syndromes, these cancer syndromes drive. In a very significant percentage, the first indication that a tumour was arising in the context of a germline mutation of a familial syndrome came from the tumour analysis itself. It was not known before that tumour was analysed. And so you get, this is why you get such a significant, I think, new referral rate for genetic services that arise from these genomic programs. But again, that was something of a surprise for us, and I think there's more there to come. One of the other early features we were trying to work out is to what degree the various pathways, which particular alleles and pathways were driving cancer susceptibility in children. What this is showing you is a particular analysis comparing the rate of mutations in the germlines of these genes compared to the NOMAD cohort. So do we have a statistically significant enrichment in our cohort in children compared to all of NOMAD? And, you know, we see those in the genes that we would typically expect to be enriched in childhood cancers. But one of the things that was really interesting for us, and because we tried very hard to understand, if you like, BRCA-ness in childhood cancers, was that we weren't enriching for those genes in childhood cancers over the NOMAD at all. So it doesn't mean to say that germline BRCA mutations don't contribute to childhood cancers, but it's rare. And it's much rarer than imagined, and it's certainly very distinct from contribution to adult cancers. And this I'm sort of got a little out of order here, but what we're talking about here is that these are the types of information in a tumour that kind of led us to understand the information that we were dealing with a germline derived tumour. And this manuscript is under review currently. It's been rejected once, but hopefully it'll appear. But this is an important message. And just for those of you to emphasise the value that RNA sequencing of the tumour for a small number of patients made a significant influence on our call of a germline the pathogenicity of the germline variant, particularly where, for example, we see the germline variant leading to loss of expression of the tumor suppressor, or we can tell the consequences of a splice variant in the RNA. So those sorts of things help us enormously. So this one here is just to tell you a little bit about the future of this. This curve here represents the number of patients enrolled to 0 up in this state, up to this state. And the red here represents the number of known cancer risk alleles that have been diagnosed, as 16% to date. This represents the number of variants of unknown significance in those same genes that we are unable to interpret at this stage. So whilst a significant majority of these are likely to be benign, they're not likely to be driving cancers. There's certainly some here that will be. So it's very likely, I think, that we are still continuing to underestimate the rates of germline mutations driving childhood cancers, and I suspect that's true across almost all cancers. So I've kind of said all of that, and I'll skip on now because I realise I am running rapidly out of time because I've got a lot to cover. And I'm keen to get to the biology. So the high throughput screening program, I just want to touch on this briefly because it is a point of difference for most genomics programs for us. And this was the idea that we could take tumour samples either directly from patients or that were cultured in vitro or in vivo in a PDX environment for some time, dissociate that material, expose it in a high throughput way to a panel of drugs, and from that, some genomic independent information would arise that might influence a treatment decision. We spent quite a lot of time here showing that we just didn't get responses or no responses across every tumour. So we see some distinct patterns in this data which would suggest that we've got at least some specificity in here, and you can see that we've got the tumour type ranges across sample types and different ranges of responses depending on the classes of drug that we're using. And I should have mentioned, I think I popped it, no, maybe I didn't, it'll come up. This is published in 2023 in Cancer Research, if you want to see that correlation between the different sources of material and the drug responses. But we did multiple testing of same materials, harvested in different ways directly from patients, some passaged in vitro, some through mice, and could show a consistency of drug responses across those materials, and not only by individual drug, but by classes of drug. So there's some sense here that these cultures or this screening program is replicating the original conditions of the tumour, but of course we're testing tumour in a dish, and you know that's a very different proposition for a clinician to say, well, yes, I'll pick that drug and use it. This is the workflow. We use a range of variables to call hits, and now we've defaulted largely to LC50s and scores because this allows us to distinguish not just it allows us to distinguish cytotoxic and cytostatic effects, and allows us to give some sense of what people might expect a true response to be. And when we're reporting this information, because this goes into the zero reports and it's made available to clinicians when we're able to do it, is we are providing a sort of waterfall plot to give them information about whether drugs on these particular platforms are cytotoxic, so whether they're killing those tumour cells in these assays, whether they're cytostatic, or whether they have absolutely no effect at all. Now, the limitation here is sample material, right? So if you're getting small biopsies or needle biopsies, the possibility of still having viable material that you can either directly dissociate or inject into a mouse or culture up after you've extracted DNA and RNA is quite low. And so there are significant limitations to the number of samples that we can put through this program. And so you can see here on the left, the types of samples that we've got and the numbers are there. So there's about 184 of well over 2,000 at the moment. But every week we get a small number of samples that we can take through this program. So it continues to grow, but it is limited in its reach. Its impact is most seen when we get both a genomic call for a targeted drug and a response to that same tissue in the screening. So where the screen matches the genome, that is a reassuring orthogonal piece of evidence, and that often prompts people to go for that particular targeted drug. I was trying to think about this yesterday, but I don't believe I've ever seen a clinician opt to choose a targeted drug on the basis of a screen alone, where they don't have some kind of orthogonal validation that there's a mutation that they're really targeting. And one can understand why that might be. They just don't have the evidence. For this program, it is feasible, it's doable. It can run off multiple sources of material. There are similar programs now operating out of the Princess Maxima Center. There's another one in Florida, in Boston now, I think. So there are a number of places that are trying to drive these sort of high throughput screening platforms on as a secondary level of treatment recommendation. The uptake is almost always where the molecular data and the high throughput screening match. So you could question, if you already had the molecular data, why would you do this? And I think it's a reasonable question to ask us. And we still do not know the answer to point 5 there, is what does a clinician require in order to select a drug from a high throughput screening data alone? Now, I've said it a couple of times, let's go to this one, that many of the clinical benefits that we've seen, we're going to divert to a bit more cell biology and cancer biology and science now, just for a moment. If you don't mind, you can nod off if it's not your thing. That a lot of the early clinical responses that we saw came from receptor tyrosine kinase inhibitor drugs that were available and were able to target lesions that hadn't been suspected to be in a particular cancer. So when we sequenced it, we found something that was targetable, would never have come to light with standard of care analysis. And so a therapeutic option was made available that wasn't available before. One of the important questions for us is whether we really are covering the full range of RTK activating events in childhood cancers. I put that question there because obviously I think the answer is no, and I'm going to try and persuade you of that yet. And I want to illustrate this with a couple of examples, one that's published now and one that we hope will be published soon. And it kind of has a hematological flavour. Piers, it might interest you, it might not. Let's see how we go. Many of you may know that in the last 5 to 7 years, a class of drugs called NTRK inhibitors, tyrosine kinase inhibitors, targeting the TRK genes, have become available, and they are particularly good at inhibiting signaling driven by fusions, where a 5' gene is struck to the tyrosine kinase domain of one of the NTRKs, and that's the primary driver of that particular cancer. And in this instance here, you'll see one of our early experiences. We'd found this particular fusion in this child with a huge sarcoma in their chest. They're a few weeks or weeks old. For those of you who know, that child there is intubated in ICU, was unable to be extubated. The fusion was found through zero. Treatment was started, and a few weeks later, this tumour has shrunk, the child's extubated, and ultimately that child went home on that therapy alone. Right? Now, that doesn't mean that that child's cured for life, but that's a kind of astonishing response to see, and particularly in that tumour type. So it's, even though these things are vanishingly rare, finding NTRK fusions is a really high priority because you've got an extremely good therapy to offer people who would otherwise have almost no choice. So even though they're rare, you've got to find them. They're really important. So what do you do when you find one of these? And this was, to our knowledge, the first time this had been seen. And this is an internal tandem duplication of the transmembrane domain, not the tyrosine kinase domain, of NTRK2. And this was in a central nervous system tumour. And on immunohistochemistry, it's associated with TRK staining. So that could mean that it's activating. That's not very specific. So what we do in my lab as much as possible, and it's very hard to do this in real time, is we try and clone these things from patient samples. We try and find model systems in which we can test their function and their drug response. And from that might, either for that patient or subsequent patients, come a therapeutic recommendation. So when you express these NTRK ITDs in cell lines that are dependent on a cytokine, for survival, and you withdraw that cytokine, the NTRK ITD survives. So it's acquired resistance to cell death. When you put these cells in culture without interleukin-3, they are capable of proliferation. So that NTRK ITD is sufficient for these cells to acquire those particular hallmarks of cancer: cytokine-independent proliferation and resistance to apoptosis. But more importantly, they are as sensitive to the NTRK inhibitor drugs as are the same cell lines, but transduced with the NTRK fusions. The only difference is that if you have an NTRK fusion, you can get the drug on trial, and if you have the NTRK ITD, you can't. So a patient with an NTRK ITD, unless we happen to just fib about what this structural variant is and call it a fusion, that patient won't get this drug. Now, in this particular instance, that didn't matter. And this was a one-off, this was a very rare event. But within 2 months of us publishing this particular case report, arguing that this should be part of the sort of, this should be introduced into the, as a recommendation, or sorry, as an indication, we've found at least 2 more of these, both of them in high-grade gliomas, both of them with a range of other mutations, but it's clear that this is not a one-off event, that these are recurrent. And the problem is that all the standard of care testing, that immunohistochemistry or targeted testing, will not find those ITDs. So, you know, we need to look because they are potentially targetable. And I've got more evidence here to show you. I'm sure you don't need to see an IGV screenshot, so I'll just go through it. But, you know, these are there. They're associated with other mutations in the context of high-grade glioma. So these are fairly typical mutation profiles and associated with high expression. Have I got till 12:30? Okay, I'll go. Oh yeah, I'll give you plenty of questions. I'll skip through this. This is the second example that I wanted to get to, which was about Sybil mutations, which for those of you who are perhaps more hematologically inclined will be of interest. Sybil mutations are associated particularly with myeloid leukemias, particularly when there's a FLT3 internal tandem duplication. Germline mutations are associated with JMML. The somatic, many of these are thought, some of these are thought to require the complete deletion of the wild type allele, and some act as dominant negative. So within the 0 cohort, we see a significant clustering of Sybil mutations, and particularly, John, around the ring finger and linker domains of this E3 ubiquitin ligase. And the vast majority of them are in brain tumours. They're not in hematological malignancies. And many of these are exactly the same mutations that arise in AML, but they are appearing in CNS tumours. So the question really arises is, are these things activating? Are they RTK activating in CNS tumours? And if they are, which RTKs? And if that's true, do RTK inhibitors have a role in the presence of civil mutations in brain tumours? And I'm not going to pretend to you that I know all the answers here, because I really don't. But again, we've taken this approach where we've started cloning. I'll skip over this. This is just a heat map of the range of mutations. Let's get to the real data, where we started to model these in organotypic cell lines, so in neuroblastoma cell lines and high-grade glioma cell lines, but also in our interleukin-3 dependent cell lines, to ask whether these mutations really do drive RTK activation. One of these samples, one of these patients, had a sample that went through high throughput screening before we were really cognizant about civil mutations in brain tumours. And this one sample, N of 1, just happened to have an unexpected and at that time unexplained response to a broader spectrum tyrosine kinase inhibitor, which we didn't understand at the time. And we had some of that material. So this is the patient material on the left compared to another patient's material. It also expressed that exon 8, 9 deleted form of Sybil within the tumour itself. So we've expressed these variants. I'm just going to show you that for the moment this 8-9 variant in various neuroblastoma and other cell lines. Wherever we express this variant in cell lines, we roughly halve the doubling time for these cells. So it's an early indication that this is driving proliferation or increased proliferation in most of these cell lines. And where we do a type of experiment where we starve cells of serum and then re-stimulate with particular ligands, in this case EGF, we can see that in the presence of the Sybil variant, we get sustained signaling of that receptor tyrosine kinase. So it suggests to us that in these other cellular contexts beyond AML, we're getting sustained signaling. And we see that not only with EGRF, but with the downstream pathways that are driving from that. So there's an early signal here that these things actually are potentially oncogenic within this context beyond AML. I mentioned there are a couple of types of Sybil mutations, those that delete exons 8 or 9 or both. That's the sort of common variant and the one that's most often seen. We also have a range of missense mutations that are also clustering in this ring finger domain. And so we're trying to work our way through whether those point mutations, which are more unique to these brain tumours, are also capable of transforming cells. And at least for this one, the C384G mutation, we can see that there is some protection against cytokine withdrawal-induced cell death, at least in the BAF3 model, and so we're looking beyond that. But more particularly, where we make those cells EGFR dependent, then we start to really see these point mutations driving more proliferation and protecting cells against cell death. So if we sort of shift away from that sort of less typical model of IL-3 towards something that is perhaps more associated in CNS tumours, we start seeing these point mutations emerge as truly oncogenic. So they are able to drive proliferation in the absence of EGFR and promote survival. And as well, they have those same co-stimulatory effects when you treat with EGF ligand. So these two mutations, which are novel, this delta 8, it's a sort of splicing variant, it disrupts exon 8. This particular point mutation, we believe, are activating and potentially targetable with tyrosine kinase inhibitors. We have across the cohort a whole range of splice variants that disrupt or skip exon 8 within Sybil. And at the very least, we can see the presence of those truncated exon 8 deletion within the RNA of those tumour samples. So we don't know how many of these splice variants might turn out to be targetable, but the number of mutations here is much higher than we imagined from the start. So civil mutations, I'll run through that summary for, there we are. So I would argue we're far from understanding the full range of this particular class of oncogene activation. And so we are missing a lot of therapeutic opportunities because those indications haven't been established yet. And so we're keen to see if the preclinical evidence is persuasive enough for people to go ahead. Because if we wait for a clinical trial on the basis of this mutation rate, we'll all be long dead before that trial is ever completed, because these are rare. We think they're targetable. And I just want to, expand on this point down with number 5 here. We have a class of variant in 0 we call VUS 3.8, which means we really think they might be pathogenic. We really think there's something there. It's either the gene or the recurrence or the expression of something that's in there, but we don't have sufficient evidence to prove it. Right? And so how do we explore these things at scale in preclinical experiments to make an impact here? Because it's clear to us also that our punters, our customers, the oncologists are interested in this preclinical data as well. They want to know that because it's becoming more persuasive to them in 0, that might be a basis on which they will select a treatment. There's lots else going on within the 0 program. You'll please, I'm at the last slides. Liquid biopsy is a huge investment for us, and I think that started in a very minor way. I can see Sarah Jane at the back up there that we started that. This has grown to a much more significant program than it used to be. We take the approach of custom designed panels for liquid biopsy built off the genomic information of the primary tumour itself. That's allowed us to be very sensitive, but you can imagine, I'm sure, that to do that at scale, it's a very expensive and challenging program. But that's where we sit at the moment. More importantly, I guess for people in this room and for Peter Mac, 0 is approaching well over 2,500 patients enrolled. So we believe we're capturing virtually every diagnosis in the country. We've just been funded for the next 3 years. This is funding not through MRFF or as a research project. This is funding that's coming through the health department. And we have a number of significant requirements for us in that funding. Part of it is we have to get at least the testing platform of 0 accredited. So what that means for us means we find the pathway to accreditation in 3 years' time. I don't think that means that we turn ourselves into a pathology service or a service provider, at least certainly not in 0's current context. But that's written in the agreement. We have some of that funding particularly salted away for expanding the program to adolescents and young adults. And the way that is being thought about is essentially in 2 pilot programs or 2 centres to pilot. One, we hope, is here at the Peter MacCallum Cancer Centre, because it will capture the vast majority of adolescents and young adults who have either paediatric type tumours or are relapsing from a form of paediatric tumour with the same disease. And the other centre is likely to be in New South Wales. But the way we want to do this is to pilot, to make those mistakes or do that engagement the way we did in the first place, try and get this as right as we possibly can, and then let it run and see how it goes over the next 3 years. That engagement with AYA has really been consumer driven. When we got the funding, it was a surprise to us that that was one of the requirements. So it's been heavily consumer driven. And so your patients want this. We do have more clinical trials being attached to 0. I haven't mentioned the one that's running at the moment, which is more an immuno-oncology one. So, that's my third message. I think I've hammered that in the extreme. There is an enormous number of people to thank. I'm pleased that I'm not going to read out every name, but it's a huge program. It's a national program, and there are many international collaborators as well. And of course, it's funded by a very diverse set of supporters, including a number of philanthropic organisations. So I'm happy to take questions, and thanks for listening.
Thanks. Thank you. Thank you, Paul, for a fantastic talk. Any questions? I've got 2 microphones that don't work.
There's one up the back.
Thank you very much for your talk. So yes, you mentioned really briefly at the end about the targeted panels that you'd like to be doing, or that you are doing, on liquid biopsies, and how it's expensive to have a customized panel. But have you considered, maybe you already have considered, having more of a generic panel, maybe 1 or 2 panels that have been informed by your whole cohort's mutations that you've seen? Is that feasible? Is that, what are some of the hesitations you might have about doing that instead?
No, I think it is feasible, because we will get recurrent mutations across tumour types. So that is feasible. But I think the real sensitivity here is about chasing more than 1 mutation in your liquid biopsy. The more that you cover for an individual case, the greater the sensitivity you'll have. And remember, for us, for liquid biopsy, even in the youngest patients, we might get 500 microliters of blood, of plasma to work with. We're not going to get mils. So we have to think about balancing that sensitivity and specificity, and that's one of the reasons why we want to do multiple mutations from the same tumour to track. Now, of course, we've got to get lucky and make sure we pick driver mutations and things that are not passengers. They're just going to disappear. And, you know, for most paediatric tumours, we haven't even established just how strong that relationship is between the burden of circulating tumour DNA and tumour bulk. How strong that really is. But there are some signals that it works. And one of the other interesting findings from our program is that we can pick up circulating tumour DNA very effectively from brain tumours. So you don't need necessarily to sample CSF. But yes, I think you're right. We could have a nice panel, or we could have a set of mutations that we're going after, and we can sort of cherry pick those for an individual and use them. But I think the key is to do more than 1 per sample.
Can I ask, Paul, I was relatively surprised to see the diagnostic sort of contribution to be only 5%. I would have thought it would have been a more powerful way of resolving diagnoses and clarifying diagnoses. Is that something you want to comment on?
I think in the first instance, so remember that was from the first study, and I think that's changed because now we're getting many more cases come in where the diagnosis is actually uncertain. And so the molecular features and the methylation help resolve that. So that percentage has gone up. There's a particular cohort within the Zero study called the rare cancer cohort. Very often those diagnoses are unclear, or at least, uncertain at the time. And that sort of runs as a separate study, and we're able to resolve those in most instances.
And how do you get the drug? So it's is it sort of beg, borrow, and steal sort of phenomenon, or do you have proactive relationships with pharma? Or
so within Zero, we've appointed someone who really works as a direct liaison between the program and pharma, and helps clinicians in each centre engage with those various drug companies to try and access the drugs. So we've tried to sort of build in within the Zero program a champion who helps those clinicians access those drugs. Some clinicians already have very good pre-existing relationships and do well at accessing the drugs, but the bottom line is that it's still a challenge. So there is a lot of beg, borrow, and steal in there as well.
John.
Paul, very keen to see in your cases here, especially when you're targeting a lot of TKIs. Are you doing much follow-up and relapses and looking for resistance mutations in these cases?
Yes. So We are now open to relapse, so we've now got an increasing cohort of samples where we've sequenced at multiple stages across that treatment journey, and we definitely are seeing the emergence of RTK resistance mechanisms, some of which you would anticipate, mutations in the tyrosine kinase domain, and some which are completely unanticipated. So the emergence of incredibly complex genomes that have just simply escaped really all treatment, some things that you might expect, chromothripsis and genome shattering events going on. So not necessarily at point mutations, but I don't think we have enough yet for a separate analysis, but we are able to get and sequence those. And particularly as the number of patients who actually get the targeted therapy go up, we're going to get more understanding what the resistance mechanisms are going to be. Yeah.
Can I give you one comment about the
Yes.
Sybil?
Yes, tell me about Sybil.
No. So in hematology, there's actually been investigated giving ponatinib for these patients because it's assumed that it's essentially degrading FLT3.
Yeah.
And so we target FLT3 through ponatinib. Now that's parochial reason. That's not necessarily the best way of targeting FLT3, but it may be, like I was listening to say, it may be such that it's doing something different in different cellular contexts. It's EGFR in that context, and it's FLT3 in AML. And the other comment I was going to make was about NTRK.
Yeah.
We saw an NTRK fusion in a patient with AML and gave them larotrectinib and did essentially nothing. It kept it at stable disease. Obviously pointing out that adult and pediatric cancers are likely to be different and cell dependent.
Absolutely. And I think there are newer classes of NTRK inhibitors, some of which may be more effective. But, one of these weird resistance cases that we saw where we didn't get a point mutation in NTRK2, we just had a genome that was just, 1,000-fold more complex than the one we were originally treating, was in a patient who'd received an NTRK inhibitor.
Right.
I think there's a lot to learn about these, both with resistance and response. But Sybil and brain tumours, right? It's
amazing. I'm a list to send you all our Sybil variants that were detected.
What we're going to attempt to do is try and sort of do a kind of saturation mutagenesis across that region and try and understand that, because that way we can get ahead of the curve, try and do all those deletions and then understand, well, when we see it prospectively, okay, we know what this one means.
Yeah. Long reads at all? For people online, that was anything about long reads at all. With a smirk.
Part of the Zero extension program, so the next 3 years, is about research and development and understanding the impact of newer technologies, which definitely includes long reads. So we've got engagement with Oxford Nanopore. I can't sit here and say that I know what we'd use it for over and above now, or what the use case scenario would be. I'm sure we'd be a lot better at phasing variants. We'd be a lot better at understanding some of those complex events that we try and resolve with shorter read RNA. But I think we hope to learn. That's all I can say. We haven't really done it yet.
Just a quick question. I was just curious with the liquid biopsies.
Yeah.
You mentioned you had a lot of structural variants. Can you even pick them up with the short fragment size, or are you just going after point mutations?
We've been doing work to try and identify structural variants, particularly fusions, for example, through individual breakpoints, right? I don't know that I can give you a clear answer to that. I'd have to probably default to either Mark Cowley or Rob Salmon to tell you the answer. I know we've been trying. I can't tell you how successful it is.
Okay, further questions will have to be asked over all the treats outside in the foyer that I've been asked to remind you about. But it just leaves me to thanks Paul Ekert for a fantastic talk, and thanks for coming along. And
I look forward to all being in Zero next year. Thank you.